Alright, so making this video for the photosynthesis lab. So the photosynthesis is basically the opposite of the previous lab, right, which was cell respiration. Cell respiration, we are burning glucose, right, metabolizing it, and it becomes CO2. Plants use photosynthesis to do the direct opposite. But we'll talk about this in class as well. So plants are able to take CO2, which is a gas, they fix it to become carbohydrate. CHO stands for carbohydrate. Glucose is an example of a carbohydrate. Photosynthesis requires water and CO2. It literally fixes CO2. So fixing CO2 means that it goes from being a gas state to a solid state like glucose. And everything in the plant that contains carbon, right? The carbon that's in the lipids, that's in the proteins, or in the carbohydrates, the carbon comes from CO2. So you say that's its carbon source. So the way plants absorb CO2 from the atmosphere is they have little holes in the bottom of the leaf. So this is a cross-section of the leaf. A leaf is an organ of the plant. It's the site of photosynthesis. And photosynthesis actually takes place in the mesophyll cells, right? The chloroplast of the mesophyll. So the sunlight has to get through the leaf, the surface, and reach the chlorophyll found in the chloroplast. So a cross-section, this would be the top part of the leaf, this is the bottom. The top has the upper epidermidis, the bottom has the lower epidermidis. On the top of the leaf you have palisade mesophyll cells, and then below them are the spongy mesophyll cells. There's actually air spaces in between those cells to help get the CO2. So CO2 can enter the leaf through the stoma. Okay, it's a little pore. So this is, if you're looking at the bottom of the leaf, and you can see the guard cell on each side of the stoma, right? There's a guard cell here, there's a guard cell here, there's a guard cell here, there's a guard, and then in between, there's like an invagination. You can open and close the stoma via the guard cells. They can close it or open it up. So if you have to allow CO2 to get in, but plants make oxygen, so the oxygen has to get out of the plant through the stoma as well. The other kind of problem they have is water, most of the time, is leaving the plant through the stoma. So those are the three gases that come and go. You know, CO2 goes in, oxygen and water mainly go out. Okay, so so you might see this on the practical. You should be able to label the parts of the leaf, right? This is the leaf, the cross-section of the leaf. We would have looked at it in the microscope had we had done this in person, but that can't happen. So one of the experiments we would have done in lab was to put what's called cobalt paper, it's blue, before you put it on the leaf. So this is the top of the leaf here. This piece of cobalt paper is on the bottom of the leaf. And then we would have given it some time, you know, say the length of the lab, and then check these pieces of paper. The top of the leaf, no water gets out of the leaf that way, right? So the pores, the stomata, are on the bottom or the lower epidermidis. So this would turn pink from coming in contact with the water, and the other ones that we put on top of the leaf would not turn red because water doesn't come out of them at the top of the leaf. So it's a pretty straightforward. So in paper chromatography, there's keratines, xanthophyll, chlorophyll A, chlorophyll B. I put them on the board. Hopefully you can see this. Keratine, xanthophyll, chlorophyll. There's an A and a B chlorophyll. There's not much difference between the two, but they're two different molecules. These are plant pigments. So in the leaf, Pigments serve to absorb light. That's really the definition of a pigment, right? When a photon of light, that's you know, the amount of photons that leave the sun are incredibly high numbers. They're constantly coming and or when we turn the lights on in a room, the light is a release of photons. Okay, so when a photon hits these pigments, the photon is basically providing energy so that the plants can use that light energy for photosynthesis, right? That's how photosynthesis occurs. You need an input of energy to make CO2, turn it into carbohydrates. And we're going to get into all the details of that in class when we do chapter 10. Okay, so I'll explain that process. This purpose of this experiment is to separate the pigments, right? They're all together inside the leaf. So what we'd do in the lab was we'd have taken a small chunk of leaf and crushed it all up. And then we'd add acetone to the crushed up leaf, which would extract the pigments. The acetone would turn, it's basically a green color, because most of the pigment is chlorophyll, which has that color. Then we would we take a drop of the acetone containing the pigments and put it on this chromatography paper. So it'd sit in the bottom. We'd add an organic solvent to, let's see, let me add some. So we'd add an organic solvent to the bottom of this tube. I'm just using a red color so you can see it better. And then we'd add the paper, and then at the bottom, right, the pigment or the organic solvent would move up this paper, and it would carry the pigments that we added at the bottom. So when you see this depiction here, right, the pigment started here. Chlorophyll B moved up to this part of the pigment. Chlorophyll A moved into this vicinity. Xanthophyll moved further in the time period. It takes about a half an hour to an hour for this to occur. And then the carotenes, they're the smallest pigment, so they moved up the paper the quickest. So, you know, movement of the plant pigments up the paper, you know, diffusion is how they moved, right? They diffused from here, point A to point B, or here, or here, or here. So they moved, like I put over there, the arrow, pretty straightforward experiment. So that's it for the photosynthesis, right? Photosynthesis is when plants remove carbon dioxide from the atmosphere as a gas and turn it into carbohydrate or glucose, which is one of the carbohydrates. And then after it does that, then it can use enzymes to convert the carbohydrate or glucose to anything else it needs. So plants, you know, they can make every macromolecule from CO2. Okay. And then these are plant pigments. I explained plant pigments are used by the plant to grab light, and then I'll explain in class how that energy is then somehow, it's probably not know all the details of it, but that energy is captured in the chloroplast of a plant cell, right? The cells of the plant have chloroplasts, and then that energy is captured to provide energy for the photosynthetic process. Okay, so that's it for this lab.